Okay, now Swati madam will introduce their speaker. Over
Thank
to
you,
Swati.
sir. Thank you, sir. I'm Swati, myself Swati, working as assistant professor in Sri Vishnu College of Pharmacy. It's my privilege to introduce today's guest speaker, Sri Chiranjeevi Venkatesh Garu. Chiranjeevi Venkatesh sir completed his B Pharmacy in Sri Vishnu College of Pharmacy during 2006 to 2010. Com- he completed his M.Pharmacy with specialization in pharmaceutics in BITS Pilani in the years 2010 to 2012. Sir has done 6 months internship on generic oral solid formulation development at Lupin Research Park, Pune. Coming to his career, Sir worked in Biocon BMS R&D Center, Bangalore as senior associate scientist who involved in pre-formulation studies, preclinical formulation development, biopharmaceutics, risk assessment, polymorphic screening, earlier development studies for phase 1 and phase 2 studies, and XRD studies. He also rented his services in Dr. Reddy's laboratories, IPDO Bangalore. He played a vital role in developing and filing complex oral, solid, and liquid dosage forms development for several markets. Currently, Sri Chiranjeev Venkatesh sir is working as Deputy Manager in Project Management Department of Biocon Pharma Limited, who involved in coordinating with several CMO CROs in developing and filing the different products in the regulated products. With this brief introduction, I hand over today's FDP session to our speaker.
Thanks, Swati, for introduction. I hope like I'm audible.
Yes, sir, you're audible, sir.
Yeah. I will just share my screen
Yes, sir, you are audible.
yeah. So I'll just share my screen like I have the some changes to this one.
Okay, sir, you can share your
So
screen.
yeah, someone confirm like are my slides are visible?
Sir, no, sir.
No, sir.
Just I will try it one more time.
Yes, sir.
Yes, sir, screen is, sir, sir, please unmute.
Yeah, Swati
Sir,
madam.
sir, voice is not audible, sir. Sir should unmute the.
Yeah, one
Yes, sir.
minute.
Now, sir, voice is audible, sir.
Yeah, I'm just sharing 1 second the screen. Yeah, is it good now? Like both the screen as well as the voice?
Yes, sir.
Yes,
Very
sir.
nice, sir.
Yeah, thank you, thank you. So like today, like whatever the topic I selected is like playing hide and seek with the poorly tasting medicines. This is like one of the interesting what is topic like which involves like how to optimize the taste with respect to mainly with the pediatric formulations. So we can keep this section as much as interactive. So in between, if someone has the question, just stop me. Like I will be able to help you. And so different technologies will be going to discuss this one, though it seems very simple like optimizing the taste of the medicine, but it is very very complex. And believe me, like I have worked on one of the project for the almost 3 years on the pediatric formulation with no success. So this is how the complex like different techniques involve like to get the means change the bitter molecule of the taste to bitter molecule to the make it the palatable. So So coming to the contents of the presentation, today we'll go through the basic taste, like what is the taste means and the physiology of the taste, and followed by the compliance of the pediatric formulations, how the compliance rate is and what we need to do to increase the compliance. And the fourth factor is like to be factors which to be considered for developing the taste masking formulations. And next is the taste masking methodologies and how we need to select the methodology for which molecule. And finally, like to evaluate how taste masking method we approached is effectiveness in masking the taste of the bitter molecule. So most of the content I have taken from this article from the ADDR, playing hide and seek with the poorly tasting pediatric medicines. So means I recommend like whoever there in the call today, like to go through this article, and even from the whatever the advanced drug delivery review article. So they will elaborate much more on single topic. So all the information will get in one place. So ADDR is like one of the very good journal like for the reading purpose. So coming to the physiology of the taste buds, like as everyone might be knowing, like so there will be the papilla. So in the papilla, we'll have the taste buds. In the taste bud, we'll having the taste receptor cell as well as followed by the sensory nerves. So whenever we take any of the material or the molecule, like whatever the food, so it will bind to the papilla, and then the receptor cell will trigger, and the sensory nerve it will stimulate and will go to the brain. And going coming to the physiology of the brain, like once we take the whatever the material food, so in the tongue, like there are three nerves majorly, like one is the cranial nerve, other one is the vagus nerve, as well as the glossopharyngeal nerve. These three nerve will stimulate the initially thalamus, followed by insula, and finally it will go to the orbitofrontal cortex, which is our the main sensory organ. And as everyone knows, like we have the different taste receptors papilla and situated across all our tongue. So the reason like why I'm discussing this here is there are some of the principles or the materials which where we can use them to block this sensation. So nerves we can make them numbness. And this is the trans taste transduction, like what is the mechanism. So if you see, there are the mainly two types of the receptor mechanism. One is the G protein receptor, other one is ion channel receptor. So for the taste sweet, bitter, and umami, G protein receptor will stimulate, and the salt and sour like ion channel receptor it will be stimulate. So in both the cases, whatever is there, like the calcium ions need to be released, and which it will trigger the receptor, and through the neurotransmitter will be released, and then it will stimulate the nerve, and it will carry on to the brain. Similarly, with the G receptor, the same like once any molecule, like whatever the food binds to the receptor, G protein will be activated, and it will be giving the releasing the enzymes, and then the secondary messengers as CAMs will be releasing, and which will trigger the release of the calcium like ion channels, and then the depolarization it will go to the nerve impulse to the brain. And this is one of the common myth like I just want to discuss, like theory of the taste coding. So there are the two lines, which is one is the labeled lines, other one is the pattern coding. So labeled lines is like if it is a receptor, like I means food is touched to the one receptor, only that taste will be triggered. So from like we might be going through whatever the literature we have, like a tip of our tongue will sensitize the sweet, and the corners will be the salty, and the end it is the bitter. So, but this is a common myth. And if you see the papilla, whatever they're located across the tongue, each papilla can recognize all the taste, but the intensity of the taste will only the differ. If you see the circumvallate papilla, only the bitter taste where it can intensize more, sensitize. And similarly in the foliate, sweet it can sense more. Fungiform also the same, like the taste, the way the intensity is more. So all the receptors we can taste means sensitize all the taste, but it is only the at whatever the intensity it is like sensitizing, it might differ. And then coming to the why we need to like mask the drug molecules, especially to increase the compliance of the pediatric formulation. So whatever the literature shares, only the compliance rate, especially in the children, it is only 11 to 93%. And major factor for it is the formulation and the palatability. So if we see, like if we increase this, these are some of the factors which we optimize, the obviously the compliance rate will increase. So for that one of the major thing is the palatability. Palatability is nothing but the overall like taste of the molecule, whatever the food or material. So it is including of all the smell, taste, aftertaste, texture, mouthfeel, and possibly like sometimes it is the vision, like how it is appearing. Like if you see some of the colored foods, like it seems to be tempted to take that food. So all this overall will resemble to them, means combined all together it will reach to the palatability. And the second most important is the swallowability. If say like for the pediatric, especially if the solution volume it is more, like the people like the kids, so they shows like reluctant no not to take the what are the doses. If it is a very small within the few drops, can be easily administered. So similarly with the size and shape, especially with the, some of the uh, uh, means older kids, where the tablet size and shape will matters. And the other one is the required dose and how much frequency we need to give and how much volume we need to give and how much tablets we need to give. So, and the other one is a selected administrative device. So instead of the taking the oral, like if it is a nasal route or like ophthalmics, so it is like very easy, like without the taste, even we can like administer the drug to the particular parcels. And the next is like the primary and the secondary container closure system. So if the remember like some of the calcium tablets which we used to get in the earlier like cases like the giraffe shape containers, so if it is like pleasing, like the even the kids will like to take that one. And similarly if the tablet shape itself are jealous, if we are getting in the different shapes like the bear, everything like what are the different shapes, if it is like seems to be colors and the promising, like the kids will obviously wants to take that. And the next is like factors to be considered during the development of the pediatric formulations. So whilst developing the formulation, one of the thing like which we need to keep in the mind is especially the selection of excipients. So based on the like benefit risk approach only, like we need to select. So out of it, like which is like the physicochemical properties, obviously the stability and the solubility of what are the excipients we're taking, especially if you want to do the solutions, solubility will matter a lot. And similarly with the compatibility, whatever with the container, whatever the filler we have taken or the glass vial we have taken. So compatibility will matter a lot. And next is the purity of the whatever the excipients or the API we are taking. It should be as pure as possible, especially for the kids if you are giving for the younger kids. And similar to the toxicity levels, if the any of the excipients like more than 50 something, it may cause the toxicity. So that should be kept in mind. So it should be based on the only IAD limits it to be taken. And similarly with the acceptable daily intake based on the FDA like IAD limits. And tolerability, this is like for the European markets, especially for the US markets, tolerability will make a lot much factor, because like they're the kids like there might be some allergic to some of the things. Even with the mannitol, some kids might have the allergies to the lactose, even like milk. So in this case, like specifically, we need to optimize like to consideration tolerability of the excipients we need to take. And then the patient age and the patient susceptibility, whether it's means if it is like for the diabetic patients, we need to ensure their sugar should not be there, and even sugar is there like a glycemic index should not increase to the higher level. That all the factors need to be considered. And the next one is the dosage regimen, like how much of frequency of administration we need to give and how small we can keep it, how minimal the volume we can keep. This all we can work it out. And then the cumulative effect with the excipients in the concomitant medications, if there are the two three medications like in the, if you're going with the fixed dose or the combination medications, like we need to take care of all the other components also. And obviously which is the final one is the regulatory status. It should be a or like acceptable by the regulatory agencies in which we are like going to market or say we are going to sales it. And this is the one of the last statement like which we need to keep in the mind. So in the adults like miniature like the kids, so their taste receptors and our taste receptors might be completely different. So if it is feeling like good to us, might not be like feel same for the kids, especially even for the adults. So one food which we might seem say it is good, like for others might not feel the same taste. So it is very subjective, and it is like it depends on person to person. A huge variation is there. This all factors we need to consider while developing the formulation. And next more important is consideration is the regulatory one. Like some of the countries, especially they have their own guidance, especially for developing the pediatric formulations. So one is the French regulatory authority. They have the study designed to determine the acceptability of the oral liquid. So we need to prove like based on their guidance, our whatever the developed formulation is acceptable in the French. And similarly, in the European region, there are the pediatric investigation plan guidelines, which we need to ensure the whatever the formulation we developed is the have the good palatability and we're able to mask the taste of the API, means whatever the component we are using. And again, the same compatibility, whatever the administrative system we use, either the bottle filler, whatever the tubing rubber, it's like compatible and nothing is leaching or extracting into the formulation. And the other most important is like compatibility and stability in presence of the relevant common foods and drinks. So in India, although it is not common, but in the like Western, like they have the habit of like mixing the drug molecule or with the formulation with the some of the common foods and then like administrating the to the kids. So one of the thing is like the desertinib, like which is the recent, like they will be mixed with the apple juice and then given to the kids. Even it is recommended in the label. So that we need to ensure with the common foods based on the pH, the our molecule is stable and nothing is there, and the stability need to be established. So any questions till this point, like before switching to the next topic? I hope I'm audible.
Yes, you're audible.
Yeah,
Among
fine.
audience, anyone, if you want to ask anything, you can ask.
Okay. Assuming no, I'm moving to the next one. So this is the like some of the taste masking methodologies like which we usually followed in the industry. So first is like the numbing the taste buds. Like I will go in detail about all these techniques. So which where we have seen the physiology of the taste. Like there are some materials which can block the taste receptors as well as the sensory receptors that can be blocked. And the third one is the obscuration of the taste, where like we'll be masking the taste with respect using the either the sweetener, flavoring agents, or viscosity. So this also will go in the detail. Otherwise, the mixing with the food. And the third one is the modification of the API at the molecular level, especially by using the salt or like varying the pH, like making the insoluble, or in some of the cases, the prodrug approach also might help. And the fourth technique is the like creating the molecular barrier, which is like whatever the molecule we have will coat with some like insoluble material, like the taste less material, so that the whatever the taste is there, like it will not come in contact with the taste receptor. Sorry, that is the physical barrier. And with the fourth approach is the molecular barrier around the API. This is at the molecular level, like at the atomic level, molecular level, like binding the or coating the molecule itself using the cyclodextrins or the ion exchange resins. Yeah. So one of the like first mechanism is the numbing the taste buds. So in this case, like we'll be using the bitter receptor antagonists. So where there is a like more number of bitter receptor antagonist is there, like it will go and bind with the receptor against, and it will not give the place for the drug molecule, so that the drug molecule will not bind to the receptor, and so the taste will not be there. And similarly, the taste transduction cascade blockers. So where the ion channel or the transient receptor potential cation channel uh, will block it, cascade the block it, so that it will not cascade to the brain. So due to this one, like whatever the unwanted bitter taste is there, so it will be there in the mouth, but still the brain will not able to sensitize. So these are the some of the molecules which are the used commonly for the bitter blocking. So one is the AMP, adenosine 5 monophosphate, sodium ions, the thermatin, and then you have dihydrocolloid chalcone, and there are some other salts and even the mercurochrome. And there are the benefits as well as the limitations for this one. So one of the limitation is this all should be done in the trial and error. So there is no complete mechanism. Even though if we approach like use this material in the formulation, it will completely mask the taste. And the other one is the limited regulatory acceptability. All these molecules have the show some action in our body. So regulatory acceptability wise, this is a huge challenge. So we need to show all these are innate and there will be no risk to our body, or it will not trigger any other thing. And the other thing is the safety and toxicology, especially in adults, like these are established, but in the pediatric formulation unknown. And one more like complaints part, like this bitter blocker, we need to be taken priorly, like before taking the formulation. So that means like the two solutions or the two components we need to give we need to provide to the patient. So one, we need to take this first, so that the receptors will be blocked, and then the formulation need to be taken. So complaints part, like most of the people might not accept this. And coming to the benefits of this one, effective at the very low concentration, especially the neoheparin dihydrochalcone, I had the experience of using this one. So even at the very small level, the numbness it will create to the tongue, so that whatever the food we taken, it will not show the taste. And the other one is like can overcome the limitations of other technologies such as the bioequivalence. If you coat the molecules, sometimes we may face the issues of the bioequivalence. The immediate release might not happen. So in this case, that problem will not be there. And it is like compared to the conventional techniques such as the sweeteners and flavors, this whatever this effectiveness of this technique is very high, because it will block all the taste. So whatever is given, like it will not able to sensitize the taste. And the next technique is the obscuration of the taste. This is very common technique which is used for most of the like 90% of the formulations which have. So we used to dump like more number of the sweeteners, flavors, which will make it like the smell wise, so that the bitterness, if it is there, like it can be masked. And the other one is the viscosity modifier. Especially we use some of the gums, especially in the solutions or the what it is. So as if it is the solution is more viscous, and it will easily slip from our tongue. So the contact time between the receptor as well as the taste of the drug molecule is very less. So using this technique, like we'll go with the increasing slightly the viscosity of the formulation, so that it will be easily slip from the tongue and the contact time will be decreasing. And the other drawback from the viscosity modifier is like it should be optimized. If the viscosity is more, it will stay more on the tongue, and it will like leads to the more bitterness of the molecule. And as I said, this is the widely used whatever the technique. So it is highly tolerable and highly acceptable. And this limitation coming to is this is also there is we can't predict like this will work for sure. So all this is like the trial and error. We need to sit with the whatever the masters, especially who are trained very good in the tasting the molecules, and we need to get check from them whether it is working or not working. And the other one is like the bitter sensitivity of the APIs. There are some of the APIs, even at the very, very minute low concentrations, they will cause the bitterness. So in these cases, like this sweeteners or the flavors will not work, because a very minute quantity of the API will cause like more bitterness. And sometimes sweeteners, there are some sweeteners which they cause the bitterness after the taste. So that also need to be considered. And coming to the sweeteners, there are the two categories we can broadly classify. If you see this graph, there are some sweeteners which will cause the sweetness at very minimum time. Within the nanoseconds, we can feel the sweetness. But similarly, they will diminish. Like within the next fraction of second, the sweetness will go. And there are some set of other sweeteners where the effect will be the slow, but it will be the long-lasting effect. So whatever the technology usually used, the combination of the both the sweeteners will be used, especially the saccharin sodium and the SS-alpha potassium. So the initial if we take it within the nanoseconds, we can feel the sweetness, and we'll be using the other sweetness which will cause the prolonged acting. So that will keep the sweetness intensity more. So one of the things is the aspartame, neotame, and the sucralose. But as I said, the drawback for the aspartame and sucralose is the bitter after the taste. And one more technique like which we can commonly use the use of the sugars, especially the xylitol or erythritol or the mannitol. Even though the sweetness intensity of this is low, but this will cause the cooling effect. So which is the like mintness type. So that even that cooling effect will also like sometimes able to help to mask the taste, the molecule. So as I said, like all this quantity need to be within the acceptable daily intake based on the IAD limits of the FDA. So these are the some of the taste sweeteners which are commonly used in the industry. And for each sweetener, if you see, there is like a different like multiple times. So out of this, if you see, neotame is one of the widely used. So 1 gram of the neotame is equivalent to almost 7 kg to 13 kg of the sugar, which is sucrose. So that much sweetness intensity it will give. So even if it is 1 mg, 2 mg is there, we can feel like it's taste, like by opening like a pouch of the neodymium, like even like we can feel the sweetness of it. And there are some of the thing like isosulfame where it is the sweetness intensity is very less. So if we are going with the isosulfame, like the more quantity of the sweetness we need to use. So depending on the tablet weight or the tablet like a composition, like the formulation, we can select any of this one, like depending on how much the sweetness index is there. So accordingly, if we want to have the low quantity, so more sweetness index we need to use. If we can accommodate more quantity, so less sweetness index we can be used. And coming to the flavors, although the flavors will not make any taste, but it will give the feel like it will help to mask them. Means we can forget that bitter taste if something like an intense flavor is there. So our mind will be there on like sensing that taste, like instead of like feeling the bitterness of the molecule. So these are like some of the common like based on the literature. If the molecule is acidic, so lemon, lime, or the grapefruit, like the sort like taste will be mostly we can use it. Similarly, if the molecule is alkaline, like we can go with the passion fruit or the caramel. This is the different flavors which we can get it. So for the bitter flavor, usually the chocolate or the coffee or the raspberry we can use it. And similarly for each type of the taste, these are the suitable flavors that it was defined, like, which we can give the better result. But there is no hard and fast rule like we need to use only these flavors for this type of molecule. All this is a trial and error. We can sometimes we can use the mix, banana lime, banana lemon, something like whatever the mix, vanilla, cherry, even the combination also we can use it. Altogether, like the palatability need to be increased. And whatever the marketed formulations is there, this is one of the like literature compilation has done, like for anti-ulceratives, like a lemon fresh or the balsamic blends will help most. For the laxatives, cherries or the raspberry berry flavors will help most. So similarly for each type of the product, there is the different types of flavors was given as a guidance. We can start initially selecting this one, but over a period of time we can like switch to whatever the flavor is there. And the selection of the flavor will also depend on the like the population of the country. So if you see like one of the product which I used it like the Russian population, so those people like mostly the vanilla or the banana flavors. So they are like if the whatever the flavors they like, if it is there in the formulation, the palatability or acceptability of the formulation will increase. So that also can be kept in the mind while they are like designing the flavors. And the third technique is like the modification of the API. By we can change the physicochemical properties of the molecule so that we can make the insoluble the API, so that it will not able to tongue will not able to sensitize the taste. So one of the common technique used is to for modification of the API is altering the pK, means based on the pKa pH of the formulation can be optimized. So when the drug molecule is in the solution, then only it will able to bind to the receptor, and then only the taste will be triggered. If it is insoluble form, like the chances of like triggering the taste is very low. So considering the pK of the molecule, we can optimize the pH of the formulation by increasing the 2 units plus or minus, so that the drug molecule will remain insoluble in our formulation and it will not trigger the taste. This is one of the very simple technique which very easily we can follow it. So by making the soluble molecule insoluble, we can reduce the rate of the dissolution in the saliva, but in the stomach, like it can solve less, and so that the bioequivalence can be will not be altered. Our aim is like in the saliva range, pH 6 to 7, or this one, like our molecule should be insoluble and the taste should not trigger. And the other approaches commonly used is like the making the salt or the co-crystal or the polymorph, like different polymorph with the low solubility. And there are the limitations for this one, like if there is a we go with the co-crystal or salt, like altogether it will leads to the new API and which will trigger the clinical studies. So this whatever this approach, salt or co-crystal or the polymorph, this will be very limited because the whatever the innovator is there, they will go with the stable polymorph. If we are changing the polymorph, they might be trigger the even the stability issues. If it is like a metastable polymorph, again the stability of the molecule will be compromised. So there are some drawbacks with using the salt co-crystalline polymorph, but there are some of the molecules, even the diclofenac or ibuprofen, like whereby changing the salt form or the polymer form, there the where the success is there. So this is some of the challenges as discussed, like if the insolubility of the molecule is altered, there might be the problem of the bioavailability as well as the bioequivalence. And the other one is a low taste threshold. So we can't guarantee like 100% our drug molecule is insoluble, like we when we approach this approach. So for the very intense molecules of the bitter, even the very minute quantity, the nanograms, that will trigger the taste. So in this case, like the modification of the API might not work. And also there are some cases like where this approach may also show the aftertaste, like whatever the material, like the drug molecule remaining in the tank by even the minute quantities. So that will solubilize over a period of time, and it will lead to the aftertaste. And one other factor is the mouthfeel. So if the molecule is insoluble, if it is precipitated, if it is a coarse, like we may not feel like comfortable to take it. Like if it is a coarse, like our mouth might become rough, where the uh, overall like mouth feel might be the different, are very difficult to accept it. And by changing the pH system, like say like if it's the acidic if we move to the acidic side, so it might lead to the corrosiveness of the tank. And there are also some cases like whatever the other preservative system we used it, like that effectiveness might also go. So that also need to be keep in the mind while like developing the formulation. And the other drawback is, like as I said, like with the low pH, the corrosion it might cause, and it might lead to dental caries, like where the which is the like kids might not accept it, even the what are the doctors might not prescribe it. So in summary, like modification of API solubility should be beneficial technique, but it should not cause the other uh, limiting means problems. And the fourth technique is the like molecular barrier, which is the commonly used technique, where is the ion exchange resin. So everyone, like you might be aware, like there might the cation resins as well as the anionic resins. Based on the whatever the PK or the nature of the drug molecule, we can select either the cation or anionic resins. So intention of this one is like drug molecule will go and attach to the resins. It will stay in the insoluble format, and it will not be enter into the mouth solution. It not solubilize when the means the formulation is there in the mouth. And once it enters into the gastric environment, uh, it will like ion exchange resin, like in presence of the acid. So the drug molecule will be released and the bioequivalence can be achieved. So this technique is mostly used in the ODT formulations. So where the drug molecule is solubilized in the solvent along with the ion exchange resin, and it will be sprayed, right, and then it will compress to the ODT. So this is some of the like ion exchange resins which are currently available in the market, and based on the nature, we can classify them into the strong acid cation or very weak acid cation. Similarly for anions, like a stronger, weak, we can do it. And depending on our drug molecule nature, counter ion, we can select whatever is there. And right side table, this is some of the approved drugs, the drug name, and for which excipient we used, the resins were used to achieve this one. As I said, it like ionic character of the API should be opposite to the, of the ion exchange resin, so that the anion cation interaction should be there. And the resonate should be stable in the drug formulation, whatever the pH we are maintaining, either in the means solution or suspension. If it is a tablet, like stability might not be an issue. And in the pH of the saliva, pH 6 to 7, it should be stable, and the drug should not be released from the ion exchange resin. And once it enter into the stomach, the molecule should release, and bioavailability should not be compromised. And the other technique like we can use is the cyclodextrins. So like as everyone like aware, like there are the three types of the cyclodextrins in the cup form, like alpha, beta, gamma, depending on the number of the glucose molecules. So whatever the inside of the drug is the hydrophobic cyclodextrin, and the peripheral is the hydrophilic. So if you want to go with the solution formulation, this technique we can use it. So where we can bind the drug molecule inside the cyclodextrin, and as the outside of the cyclodextrin is hydrophilic, it we can go and solubilize within the solution. And once it enters into the stomach again, it should be disassembled from the cyclodextrin, and it should able to absorb it. So again, the drawback of the cyclodextrin is like even if the minute quantity of the drug molecule is not able to bind it, it will trigger the bitter taste. And depending of our molecule size, we can select any of the cyclodextrin. And even the optimization of the cyclodextrin or preparation of the cyclodextrin is one of the biggest challenge. So where we need to ensure entire molecule need to be uh, bind to the complex should be formed, and the complex should be enough to release the molecule once again. So this is some of the drug molecules where this approach was used, like which the cyclodextrin, and how much ratio is there. Optimizing the ratio is one of the complex things, and again the how the formulation is prepared, and taste masking properties, how it was given. So these slides I have circulated, even the article from which I have taken this was given in the slide. Like if someone wants to go back and we can check it, what type of cyclodextrin can use for any of the molecule. And the last approach is applying a physical barrier on the API or the dosage form. So in this case, like usually two techniques we'll use. One is the polymer film coating, other one is the lipidic systems. So our intention, if it is the immediate release tablets, we can go with the polymer film coating. So where the drug molecule we can solubilize and spray on top of any pellet. On top of it we can polymer coating, we can do it, so that like a drug molecule will not come in direct contact with the taste receptor in the while it is in the mouth, and so that taste can be masked. So different techniques can be used for the polymer coating. Most commonly used is the Brewster coating, like the fluidized bed system, and other one even the granulation spiralization or the HME or microencapsulation can also be used. Uh, but one of the drawback is like we need to disperse the solution in aqueous suspension if you want to with the fluid bed system. Otherwise, the solvent-free process such as the HMU can also be utilized. And the other one is the lipidic system. So if you want to do the controlled delayed release formulations, even the granules in the suspension, if you want to suspend, or the tablets if you want to do it, so the lipidic system can we can optimize where the release of the drug when we low. But only one of the advantages is the process wise it is simpler. There is no need of the solvents, and there will the uniform application rate of the lipid on the drug molecule. So again, this is some of the what are the polymers usually used, and this is the drugs. So one of the common polymers used is the udrazit one, where this is even used for the enteric coating formulations, as well as the opposite of the udrazit L is udrazit EPO, which it is soluble in the acidic media and insoluble in the basic media. Even this we can utilize it, and there are some of the ethyl cellulose formulations as well the HPMC. So depending on our release rate of the molecule, we can optimize use any of the HPMC grades or ethyl cellulose grades, or udrazit we can use it. And for the lipidic coating systems, mostly we'll be using the glyceryl diestyrate as one of the things. In some cases, like the wax or the cetyl alcohol can be used, where the drug release will be the very low. So, any questions on this taste masking concept, or can we move on to the next one? Yeah, I assume no. Moving to the next. So this is one of the flow chart which we usually use for which type of formulation we need to go. So if the dose flexibility is required in case of the as a children, especially if the paracetamol is there, depending on the weight, we need to administer it. So in that case, like doing the tablet or the capsular suppository will not work. We should go with the either the solution or suspension. So criteria is the dose flexibility is required. We need to assess like the acceptable API taste. If it is S, it is whether it is soluble or insoluble. If it is soluble, simple solution we can do it. If it is not soluble, we can do the suspension. But challenges with suspension is the particle size we need to optimize, and the mouth feel or the grittiness should not be there in the mouth. If the taste is not acceptable, then we need to do with the some of the techniques like the sprinkle systems or the mini tablets or the mini pellets granules. We can do it and then suspend into the suspension, and then we can market it. And the other one is where the dose flexibility is not required. We can go with the oral solids. If the taste is good, we can go with the chewable tablet or the ODT tablet directly. And if it is not good, like then we need to consider for the other techniques, like which is the polymer coating or the lipid coating or the altering the form of the API, and then we can go to into the tablet form. So simple for the adults especially. So film coated tablet we can be used it like where we can compress the tablet and coat with some of the sweetener or the film HBMC polymer, so that it will not come into the contact with the mouth. So this is all the techniques which we have discussed. Then we need to evaluate whatever the technique we used it, whether it is effective or not effective. So one of the common technique we can feel is like based on the like expression of the either the children or kids, we can identify whether the formulation is acceptable or not acceptable. If it is the good, like we can see based on their expression. If it is not good, like the people or kids might start crying or like they're not accepted. So this is one of the simple technique for the generic formulations especially we can use these techniques. But for the NCA molecules, as it was not established still in the human, so this technique we can't use it. But for the NCA molecules, whatever the technique we used, it's the electronic tongue, E-tongue, like everyone might be aware. E-tongue and the E-nose, so these are two common techniques which we will be using for the NCA molecules where the safety profile is not established in the adults or even in the pediatric. But E-tongue and the nose have its own challenges. So especially in India, like they currently very few companies have the instruments, and even the service is not good for them. And means optimizing the method or developing the methods itself is a big challenge for the E-tongue especially, and maintaining the probes is also one of the challenge. And the other technique we can use is the taste evolution studies. So this is one example I have taken it like this based on the taste panel studies, how the study will be conducted. So I will just go through it. So in this case, there are like seven formulations with the different techniques or like whatever the composition we used it. And we in this case we need to select the first the population or the individuals like and then we need to divide into the different blocks, and in each block we need to give the formulations in the random order like for each, because like if A is tasted first, some they might feel it is good, but the other one if we given in the other sequence after the G, the same A might not be the good. So as far as possible we need to approach the randomization technique and give to the people at the different intervals. So first, like in block one, if for the individual, if you're given the A formulation, we need to given the questionnaire like which will show in the next slide. When we should give them the sufficient time like to fill that one, and we need to ensure like the taste of the A is completely gone before giving the taste of the B. So one of the common technique used is like in between the formulations, so around like 5 to 10 minutes time will be given, and the puffed rice, which we'll call as the murmura, so that will be given, which is a tasteless food. So that like whatever the taste of the earlier formulation will be completely removed, and the new formulation can be given. So this is the sequence, usually the protocol which will be usually followed. And this is the different scales. This is like one of the example I have shown it like, but the scales we can optimize, the numbers we can optimize, and additional parameters also can be optimized. So this whatever the given like seven scales as it's taken, which are the hedonic scale for the sensory evolution. So one we can ask the individual to rate the taste whether it is a good acceptable. If it is a bad, either it is a one, two, a very worst or acceptable. So for each bucket, they can rate it taste, order, similarly the bitterness, as well as the sweetness, say how it is, and the sweet aftertaste. In some of the formulation, like a bitter aftertaste, even of the bitter aftertaste, after 2 minutes, the individual able to feel the taste, or within the 2 to 1 minutes, or it is less than 1 minute. there is no aftertaste. Similarly, for the sweet also, whether it is very sweet. So there are the individuals where the too much sweet also there will be not liking. So for them, like it should be the mild sweet. So that's also should be taken into consideration as a mild sweet. It is a normal okay sweet or extremely sweet. Similarly, the sweet aftertaste. So after how many minutes still they will feel the taste in your tongue. It is going within the minute or there is no aftertaste. And the final one is the overall acceptability, considering all the taste, odor, bitterness, or after sweet. How good is how bad, like whether acceptable, very good, common. So we can ask the multiple individuals, and the number we can keep as for means as number of more volunteers is there. So more number of volunteers, like more statistical analysis, we can do it, and we can optimize the formulation. So once we have the data, like there are the statisticians, like which usually the different techniques. Like one is the frequency counts. We can use it, like how many people used means told like there is a good formulation. How many people so this of sweet is optimum. How many has told like based on the frequency tables. There's a simple one we can use to judge the formulation. And there are similarly the non-parametric analysis like it done test all the critical values or the Spearman correlations are there. Like we can correlate, we can which will help to judge the formulation. So these are different techniques which can use it, and different flavor and the sweetener like vendors are there, and each vendor like in their companies they will have the taste what you can say tasters will be there, like which are the like highly paid people, and within the simple like if we give the 4 5 formulations they can able to judge very good and give the result. So what is good, and in that one what went wrong, what went good, that all report can also we can get it. So this is where like how we can judge the effectiveness of how the taste masking or how weak is the acceptability of the formulation is there. We can judge it. So that's all from my end. Like if there are any questions, queries, I will be happy to take them. So open to the forum.
Among the audience, if anyone want to ask anything, you can please ask questions.
I think like even the academic project can also be considered like any of the bitter test can be taken, and like it even helpful to publish any of the article. So in that way, like in the college, something can be planned like using different approaches. Which approach we can mask the more the bitterness of the molecule.
Sir, I am Darshan and I am from Bangalore. I have a question.
Yeah, please go ahead.
I would like to know how can personalized medicine concepts be applied to pediatric formulations to tailor treatments based on individual patient characteristics and needs.
Personalized medication, as far as I know, based on it, like for the pediatric, it will be the very complex because even it was not established in the adults. So if you see for any drug molecule, like first it should be like entirely screened in the adults. Only at the later phase, it will go for the pediatric. But as far as my understanding, like the personalized medicine for the pediatric might be the difficult.
Okay, thank you, sir.
Yeah. Thank you. So there is one. How can we know the compatibility between the API and auxiliary substance? So as I said, there is no technique. It should be the trial and error. So for the new molecules, especially E-tongue approach can be used it. So which is the like safer technique before establishing the safety profile in the uh, what are the humans. So in the E-tongue, there are the different probes. Like for each taste, there is a probe. So if it the binding way, if it's happen like that, it will trigger the response. So based on that, like probably we can able to identify. Is it suitable to use the high potent sugar? Uh, Sugar sweet music, as I said, like new time, like which is the highly one of the thing like 7,000 to 13,000 X, like in one of the formulation I used it. We can go ahead, but as such, sugar we should not use it because there might be the some of the cases for the diabetic kids where the glycemic index will rise. So most of the cases, sucrose to some extent they will be used, and mostly will go with the artificial sweetness where the glycemic index will not increase. And the next, how do the physiological difference in the gastrointestinal tract of a children impact the biopharmaceutical performance of pediatric formulation? How can this be addressed? Uh, Physiological wise, like there might be the differences, and as for the new molecules, like only the clinical trial is the only thing like which is able to address this one. And coming to the in silico, like if you people are aware that there is a gastroplay software, so in that one the pediatric module is there. So we can develop the module means model like and then we can able to fit and check it how fast it can absorb in the kids. So in silico wise, the gastroplace is one of the best technique, but developing a model in the gastroplace is also has own challenges and the limitations.
Okay, any other audience? If you want to ask anything, you can please ask.
Yeah, if not, like probably we can conclude.
Hello,
Yes,
sir.
sir. Hello. In some methods, hello,
Yeah,
sir.
yeah, please go ahead.
In some methods, where actually the physicochemical properties of the drugs they get changed. Then at that time, have you again calculated the dose of drug or API?
No, physicochemical properties will not change as long as the polymorph is same.
Uh, no, as such, in salt formation methods, okay, the solubility get increases, might
Yes.
be or might be decreases. Then at that time, what should we wait?
So
Or
that's
ionic forms like methods.
so whatever I was telling, like for the taste masking, like our aim is should be like in the whatever the mouth, the oral cavity, it should not be soluble. It should be insoluble as far as possible. Okay, and if it goes to the stomach, like it should the bioequivalence should not be disturbed. In case like if we whatever the question you ask, like the if the molecule like means based on the form change, if it is more soluble, how we need to adjust the drug? Is that right?
Yes, sir. Yes.
Yeah.
My
So while
question.
developing the any molecule, if you see like the toxicity level will be at the very high when compared to the whatever the dose we'll be taking. So
Hmm,
while developing the NC molecules, there will be some studies like all the polymorph studies will be taken care. So to the certain extent, like the dose, because uh, say suppose if it is a 5 mg, the range will be established till to the like a 10x, 50 mg or 100 mg. So where there is a no impact on the like physicochemical means bioequivalence. Even
right.
if you see the bioequivalence studies, the limit is there for the 80 to 125. So within from individual to individual, that will vary the absorption based on that one. So even if
Hmm,
the solubility based on the polymorph, either it is increase or decrease,
hmm,
uh, it should have established during the clinical trial itself. So
okay.
as far as like mostly once the polymorph is established, so same polymorph will be utilizing it. And they, if it is the difference with respect to the salt,
Hmm,
it
hmm,
will trigger altogether the new API where the clinical study need to be done to establish the dose range.
okay. Okay. Thank you. Thank you so much, sir.
Thank you.
Sir, I have a question.
Yeah.
What role do nanotechnology and microparticulate system play in enhancing the drug delivery for pediatric formulations?
Nanotechnology, I'm not sure, but the microparticulate is mostly used because if you see the suspensions, like where the taste masking is there, like as I said, like it will be coated, like the polymer coated is there, and then the means using a Wurster coating, and then it will be suspended. But nanotechnology wise, like it is most of the academic, but in the industrial scale, means I am not seen like much. I am not sure aware much, but the nanotechnology, I'm not sure on the big scale we can scale it up.
Okay, sir.
But
Thank you.
microparticulate, we can definitely do it.
And sir, also how to navigate the ethical consideration and informed consent process when we involve pediatric patients in formulation related clinical trials.
So for this one, clinical trials, I'm not much aware, but as far as I am know, I can give you like I can say like vaguely I can answer this one. Uh, so first, whatever the safety profile is there, it will be established in the adults. And
Okay.
then like in silico simulations can be done like in the as I said for the gastroblast like it what dose can be done based on the physiology. So based on the finding those dose range, it they will be go to the pediatric. This is like my understanding, but I'm not sure in the clinical design how it can be done.
Mhm.
Hello. Yeah, I think nobody has any questions in this session from now. Thank you, Chiranjeevi sir, for your valuable time spending with us.
Thank you everyone for joining like and attending the section. All the best for everyone. Like if there is any query like anything like guidance required, I will be able to help. Like Swati is having my number.
Okay, sir. Thank you.
Yes, if we have any enquiries, we will contact you, sir.
Sure, sure. Thank
Towards
you.
Swati
Thank you.
madam, deliver the vote of thanks.
Okay, sir. I thank Sri Chiranjeevi Venkatesh Garu for enlightening us with his lecture. I thank all the participants for being part of this FDP. I thank everyone in making this program a grand success. Thank you so much, sir, for your valuable speech.
Thank you. Thank you everyone for joining and patiently attending the section.